Okay, and now I'd like to introduce our next speaker is Boris Aguilar. He's a senior research student, scientist at the Institute for System Biology. And he does a lot of work developing mechanistic models for multicellular systems. And I think targeting a lot of cancer issues and problems, right? So here's Boris.
Thank you for the introduction. Hello, everyone. Let me first share my screen. Okay. Let me know, please, if you can see my screen.
Yes, we can.
Okay, thank you. My name is Boris Aguilar. I work in the Institute for Systems Biology in Seattle. And today I would like to present about digital twins, specifically for this type of cancer, myeloid leukemia patient, acute myeloid leukemia. But first, some notes. What I'm going to present is still in progress, research in progress. So, and the results has not been published yet. But I think would be good for us to show you what we are trying to do towards developing digital twins. And I would like first to present you the team that is working on this project. Everything started with an initiative of Ilya Smilevich. He united a group of friends and collaborators and from different institutions to work on digital twins. Unfortunately, he passed away this year, but here you can see a picture of Ilya smiling as always. And in red, you see key scientists, students, and senior scientists in the Institute for Systems Biology that are developing most of the work. Juan Ronchin is a senior research scientist who is one of the leaders of this project. He does a lot of data analysis, machine learning, and knowledge graphs. Yue Shan is a postdoc who is doing mechanistic modeling. Baharter Chan is a research scientist that do machine learning. And Nick, Jonathan, Christine, Luna, and Tassin, they are summer interns that worked hard this year. And most of the results are gonna show were developed for the interns. We love to have interns. So if you are interested, of course, you can talk to me. And of course, there's other collaborators in our group, Juho, David Gibbs, Besten. And we are also collaborating with people in the Fred Hutch Cancer Center, Elizabeth Krakow and Gerald Radic, who are experts in AML, as well as Pam Becker. We also have collaborators in Finland, in University of Helsinki and Tampere University. In Tampere University, they are focusing on computational side, doing machine learning. And in the University of Helsinki, they are helping us with the medical research, providing data and helping us with knowledge about AML. First, of course, we need to define what is a digital twin. And this term has been used a lot recently. Many people are using it in different ways, in different contexts, and sometimes with different meaning. And we are afraid that by people using it a lot, we may lose the specific meaning of digital twins. But fortunately, recently, the National Academy of Science, Engineering and Medicine published a report that contains knowledge from different experts about digital twins, and that specifically define digital twins and the most relevant features for digital twins. Digital twins were originating from NASA in an attempt to improve physical model simulations of a spacecraft in 2010. And here is a quote from the report that defines digital twins. A digital twin is a set of virtual information constructs that mimics the structure, context, and behavior of a natural, engineering, or social system. Is dynamically updated with data from physical twin, has a predictive capability, and informs decisions that realize value. And this is very important. The bidirectional interaction between the virtual and the physical is central to the digital twins. So many people ask if digital twin is just modeling. It's not only modeling. It has to be communication between the model, the digital twin, and the physical system to be a digital twin. So that's one aspect that characterizes digital twins. In healthcare, digital twin is a dynamic representation of a patient, typically focusing on one disease, which allows simulating potential therapeutic plans, predicted drug response, disease progressions, and adverse events using personalized models. Here we highlighted personalized models because we want a digital twin for one patient. And this is important for many diseases because the features of a disease for one patient may be different. And you will see that later, but may be different from other patients. So one patient may need a specific treatment. And that's what we are aiming for by using digital twins in medicine. This is a figure from the report that is highlighting the interaction between the physical counterpart and the digital twin. So through sensors and data acquisition systems, the information from the physical system should flow, should inform, should be used for calibration, for instantiation, for validation of the digital twin. Of course, in the digital twin with this data, we use modeling to create simulations. We can use several types of models, including artificial intelligence models, models based on first principle, important mechanisms, and the digital twin also has a component for visualization. And all of this is used together with a person to make decisions about the physical counterpart. In the next round of the cycle, because of the decision making, the physical system changes and we need to get data again and inform the digital twin. And it goes in this cycle. And this figure is taken from a perspective paper that is described here, in which Ilya Shmulevich participated. And it's also showing the digital twin cycle, but when it is applied to cancer patients or for a cancer patient digital twin. So from the patient, one can get different data types, which are then used to inform the models and make simulations. These are included in a visualization framework, like a dashboard, all these predictions. And then these simulations and predictions are used by doctors together with the patients. So these tools can also help the communication between doctors and patients, so that together they can explore different options for treatment. And this goes into a continuous loop. When a decision is made about treatment, of course, that happen, and we need to, after some time, we need to take data and go into the cycle again. One important aspect is that for each digital twin, we can start building a data set of digital twins. So we can collect a group of digital twins that can be very useful to learn more about the disease. That's why we are calling basic science here. It can be used as clinical trials. If you want to select specific characteristics of a population, you can do it in a set of digital twins. It can be used to do some population-based studies as well. And the predictions are about that can help adjust the treatment, monitor response, and track lifestyle modifications, the effect of lifestyle modifications. This picture is show you again the digital twin cycle. And there are several types of data that one can extract from patients. Some of them are routinely obtained in hospitals. But all of these data are used for models, for calibrating models. Through simulations, we can make predictions, and then together with the patient and the doctors, they define the treatments, and the cycle continues. But one important aspect that we want to highlight here is the VVUK term, which stands for verification, validation, and uncertainty quantification. This is an essential component of the digital twin that is specified in the National Academic of Science, Engineering, and Medicine report. Verification is when you make sure your implementation is correct. Validation is making sure that your predictions are according to the data. And uncertainty quantification means we need to have a quantification of the error of your predictions through confidence intervals, for example. And this is a quote from the report that says, despite the growing use of artificial intelligence, machine learning, and empirical modeling, engineering, and scientific applications, there is no standard process for reporting VVUQ and little consideration of confidence in model outputs. So the report recommends that federal agencies should ensure that VVUQ is an integral part of the new digital twin programs. So this is a component that should be part of the digital twins, which makes it a little different from just modeling. So that was a background on digital twins and how they are applied in medicine. And now we are going to focus on our specific application, which is on AML, which is a cancer of the bone marrow and the blood. AML must affect cells that are not fully developed. It means that the blood cells stop maturing. They stop at some point. So these new cells that couldn't mature, they don't function properly, and they are accumulated in the blood. And that is the kind of a marker of cancer of AML, the number of these unmature cancerous cells that are called blasts. So this disease has a dismal prognosis, and one reason that this cancer is good for digital tool implementation is that there are several data sets, public data sets, with a lot of data from AML patients. There are also a lot of clinical trials from which we can extract important data, like time courses, which are important for digital twins. And Also, patients are routinely getting data. In hospitals, they routinely get data from these patients. You can get data from blood samples. You can get data very frequently, sometimes every day. You can get the blood samples and from which you can extract certain population of cells, and that can be used for your digital twin update. And here is kind of a prototype or a diagram that shows the prototype systems that we are trying to develop. And from the patient, we can extract different data types. For example, clinical measurements, cytometry or cytogenetics. This type of data is from which you can extract different cell populations, and it's a time course type of data, which is important for dynamically updating your digital twin. But routinely, we can get molecular measurements of these patients, like, for example, mutations. And importantly, we can also get data about how the patient responded to treatment, which is very important for calibrating digital twins. We are developing a dashboard for visualization of this data and also for visualization of the predictions. And the key component or the core component of our digital twin systems is here in this green box, and is composed of different model types. For example, multicellular models. They can be agent-based or ODE-based. Here we are trying to model the population of different cell types and how they interact. For example, cancer and immune cells, stem cells and mature cells, healthy cells versus cancer cells, all of these interactions. We are also including gene regulatory network models to model the effect on mutations, important pathways, important genes. And they are directly used for predictions that can be displayed in the dashboard. But we can also use the parameters or predictions of these models to use further machine learning models to predict other properties. As I was mentioning before, there are a lot of public data sets of AML, and this can be used as well in your machine learning models. You can find data about drug screening, clinical data, molecular data, functional screening. And as part of our research projects, we are also collecting all of this data and previous knowledge encoded into so-called knowledge graphs, which is a network representation of commonly used observations in AML. So these knowledge graphs are also very informative, that can provide features and that can be used to construct models. It can be used directly in the dashboard as well. So the dashboard will include prediction of progression, prediction of drug response, and prediction about adverse effects. And this is where we want to try to model toxicity. When you get a treatment, they kill cancer cells. It kills cancer cells, but it also kills other cells. So you want to know what is the negative effect of a treatment. So this project has been funded by NCI recently in a 5-year period project, and we are in our 2nd year of the project. And next, I will show you some of our results. First, about drug response. And here is kind of a motivation of why we need to study drug response. There is only a limited number of targeted inhibitors that are FDA approved for AML. And so predicting the drug response for individual patients is very challenging. And one of the reasons is because different patients can have many different mutations. And so in this figure, here you see the frequency of mutations. On the top are the genes that are mostly frequently mutated. And these are from different data sets, in different public data sets. And as you can see, there are many genes in AML patients that are mutated, that are significantly mutated. This is in contrast to other cancer types that have 4 or a limited number of mutations that are abundant, but they are a limited number. In AML, there are many genes that can be mutated. And that makes it difficult to develop effective treatments. This is, I know there's some part of my slides that are not showing, but this is the results of a paper that was recently published by Wan Ronchin in ISB. And here, I think it shows you the heterogeneity in mutations for different patients. Here are the most common, or the genes that have mutations. I think they are sorted by frequency of mutations. And each column here is a patient. And you can see that one particular patient have different mutation profile than the other. So it's very heterogeneous, the population of mutation profiles. And in this plot, here in the y-axis, you see the different drugs. And in the x-axis is a measure of how sensitive patient cancer cells are to the drug. The more negative means that these cancer cells for these patients are more sensitive to the drug. And the positive means they are resistant to the drug. And the point here is that there are many drugs that falls closely to the center. So you cannot really define if they are resistant or sensitive. And also the confidence intervals are very, very large. So the point is here, it's very hard to determine if a specific drug will work well in a given patient. And here are results of the paper showing you some basic statistics of the relationship between mutations and drugs. This is a volcano plot, and in red is showing you the mutations that are resistant to a drug. For example, TP53 mutation, patients that have a mutation in TP53 are more resistant to cytarabine, which is a commonly used chemotherapy drug. These are more resistant to patients that doesn't have this drug. And in this figure here, it's the same information, but it's presented in different ways. And you see here, you see TP53 and cytarabine, and you see that there are many patients that harbor or have this mutation that are resistant to TP53. But there are other mutations that are related to different drugs. For example, patients that have KNRAS are very sensitive to drugs that inhibit this gene, MEK. So there seems to be some information here, very signal, indicating that we can use mutation to understand or to be able to predict resistance to drug. So more information in this paper, but this is a way in which we are trying to predict drug response using mutation data. But the data can give you so much information. We are also trying to develop mechanistic models of gene regulation to try to predict resistance to drug. And here is Boolean network model, a small gene regulatory network that contains 18 genes that are important in AML. Specifically, it contains 3 frequently mutated genes, FLT3, NPM1, DNMT3A. And we are using Boolean networks to model this small regulatory network. Of course, this network was obtained from a paper from Palma et al. In a Boolean network formulation, every gene can have 2 values, 0 or 1. And its Boolean value depends on other genes. For example, the output of OXA9 depends on NPM1. So if NPM1 is 0, this will be 1. If NPM1 is 1, this will be 0. A simple formulation, and here are a list of all the Boolean equations for all the 18 genes. This is a simple formulation, but it still can generate complex dynamics. Importantly, there are 3 nodes in the network here that are related to cancer cell phenotypes, like proliferation, differentiation, and apoptosis. These are not genes, but this indicates a phenotype of a cancer cell, meaning the cancer cell are more likely to be proliferating or differentiating, or they are likely to die. So using these outputs, we can perform simulations of the Boolean network to compute some kind of network score. This network score is the output of the model. It's defined as a function of proliferation, differentiation, and apoptosis with certain constant that we use it, that we trained. But then we can compare this network score with clinical data, as has been done in the paper. But they use some data sets. Here we are using a different data set. But in the plot, what it is showing you is the output of the model, which is the network score, and how that it correlates with the percentage of blasts. Remember that blasts are the cancer cells. So the larger the number of blasts, the more aggressive is your cancer or the state of your cancer. And we found, and this is similar to the paper, that there is a very good correlation between the output of the model, which are the scores, and the percentage of blast for different, if we consider different mutation profiles. So here every point represents patients that have this mutation, and here this dot, for example, represent a group of patients that have these 3 mutations. So we have a combination of 8 because the model considered only 3 genes that can be mutated. So you see there is a nice correlation, and the p-value is relatively small. But then, of course, we wanted to compare it with drug resistance. How correlated is the network to drug resistance? And this is for one specific drug. And here this value, when this value is small, it means that the patient is sensitive to the drug. And when this value is high, it represents that the patient is resistant to the drug. There is some signal, but the correlation is not that great, and the p-value means that it's not statistically significant. So unfortunately, we cannot predict drug response, but this opens the room for increasing the model or extending the model So that's another work that we are doing, in which we are trying to extend the model by combining two models. We develop a framework that allows you to combine different models. They should share certain genes, but the expectation is that when you added two models, you are adding more biology to your system. And doing that, you can increase your prediction capacity. And here it's the results. It's kind of similar to the previous one, trying to correlate scores with blasts, which are the population of cancer cells. And as you can see, if we use the original model, we get a poor correlation. But if we use the combined model, we can get a very high correlation. So those were our efforts to kind of improve our gene regulatory models for predicting drug response. But we are also trying to develop mechanistic models for disease progression. And this is a model that we are trying to develop, and is based on another paper of Hoffman et al. And this is modeling the population of cancer stem cells or leukemic cells. So in red, this diagram is representing the population of cancer cells in the bone marrow. Those are stem cells. And in green, the population of healthy cells. And for every cell, we consider two states: quiescent state, in which the cells don't reproduce, and also the cells are not affected by treatment, and active states, in which the cells proliferate, but they are sensitive to the treatment. There are transitions between the two states, and there is some kind of competition between cancer and healthy cells. So here are the equations of the model. This is a simple model, and we use ODE equations, or the paper uses a set of ODE equations to simulate. And this system has 4 differential equations. There are 15 unknowns, 4 initial conditions, 11 parameters. We only selected 3 parameters for calibration. And this was selected using sensitivity analysis. And here are examples of simulations of the model. And we use a tellurium to perform simulations. And in this plot, you see in the y-axis, there is this measurement of cancer progression. This is related to the population of cancer. And in the model, we have population of cancer. So we simply apply a constant factor to compare these two quantities. This is from patients, and every blue dot represents a patient measurement, and every red represents the time in which the patient received treatment and received chemotherapy. And you see that after every chemotherapy cycle, the cancer decreases, the population of cancer cells decreases. So in green, you see the simulation, and gray represents the confidence intervals. And one important aspect is that we characterize when the patient relapsed. Unfortunately, this patient received treatment, the population of cancer cells decreased, but after some point, it increased again. So let's call it relapse. And we wanted to predict relapse using these models. But in addition to predict the output that can be compared with measurements, we can obtain from the model how the different population changes. And here is output of how the other cells or the population of cells are changing in time. That can be also used to get some information about how can we recognize if a patient will go into relapse or not. Excuse me, how long, how much time I have left? I can calibrate.
You got about 10, 15 minutes.
Okay, that's good. So we are trying to optimize parameters. As I mentioned before, we focused on 3 parameters, and we establish some ranges of these parameters, and then we generated randomly several initial guesses of this parameter space. And then for every initial guess, we apply a simple optimization. And we do that for all the guesses that we selected in the parameter space, and we selected the best one. That's how we are trying to optimize these 3 parameters. But we are also exploring using Bayes approach, for which we are first, we define a prior of the parameter distribution, and then together with a Markov chain Monte Carlo model, we determine or find a posterior distribution of your parameters. And this is a simulation after we use Bayes approach. And you can see every trajectory in gray here represents one sample of your posterior distribution. This is good because you can get uncertainty quantification of your simulations using Bayes approach. This is another way we are trying to calibrate our model with the data. But what, yeah, so we also compare different strategies to optimize. If we use multiple guesses with Latina hypercube, that provides you a really good optimization because it's trying to find the global minima, but it can be very expensive because you need to run multiple optimization problems, and the more guesses you have, the better it is for optimization. So it can be computationally expensive. Using Bayes approach, we can get a quicker optimization. You can get confidence intervals, but it depends a lot on your initial distribution, on your initial prior. So that's a con for using Bayes approach, that you need to have a good prior for getting good results. What we are exploring is, and this is the connection to digital twins, is we want to calibrate the model every time we get more data from patients. So in this small video, you can see we are trying to calibrate a model here with every time we get more data, so that your parameters are changing every time you get more data from patients. And we are trying to find what is the best way to calibrate models for this type of update. So this is different than using all the data to calibrate the model. And we are doing this because we want to simulate a real scenario in which a patient goes to the clinics, get diagnosis. You don't have much information about the patient, just the information you get that day. But the patient comes in the next weeks, and every time the patient visits the clinics, you get more information. So you need to recalibrate your models every time you get more information. In this way, you are communicating patient information with your digital twin. So that's our goal here, is to find ways in which we can calibrate your models in real time. And that's how we think this type of models can be used in a digital twin fashion in the hospitals. And finally, we are also developing models for toxicity. Traditionally, when you get chemotherapy, this treatment not only kills cancer cells, but also healthy cells. For example, immune cells, white blood cells. And this can be important, if you are killing immune cells, your immune system gets weaker and weaker, and that is dangerous because you can easily get infections. So if we can predict the level of immune cells after treatment, then we can adapt the treatment accordingly. So for example, if a patient has a very low immune cells or white blood cells, which are immune cells, you don't want to give it a strong dose of treatment. You may want to stop the treatment for some time. So they go hand in hand, like the toxicity and the treatment. The treatment depends on how toxic the response was. But that's why it's very important to be able to predict toxicity. And we are developing a model that can help us predict the population of this type of cells, neutrophils or white blood counts. Those are important markers of how your immune system is. You want to have a high number of T cells. If you have a low number of neutrophils, that means your immune system is low, and that can be dangerous. So for this model, we are considering two drugs that act simultaneously. One is venetoclax, and the other is azacitidine. And we are considering also cells in the bone marrow, which proliferates. And then when this divides, the cells can mature to precursor cells and then mature to neutrophils. This process is called hematopoiesis. It happens in the bone marrow, and then the mature cells from the bone marrow goes to the blood. We are also including cancer cells in the bone marrow that can compete with proliferation cells. But these cells divide, and that can be affected by the two drugs. But the two drugs also affect the proliferating cells. So this is our basic model. Some equations of the models are described here. And this model was derived by this paper from Felix Jost. It was published in 2020. Here are some of the examples of simulations. Again, we used tellurium to generate these simulations. And in this figure, you see the count of white blood cells or neutrophils in the y-axis, time in the x-axis. And in the dots are the data from patients about the count of these cells. And the blue solid line is our predictions. And the colored bars here are the cycles of treatment, in green, azacitidine, and in yellow, venetoclax. So those are the times in which the patient is receiving treatment from these two drugs. And on the other panel, you see the blast counts. Those are the cancer cells in the blood. And the model also is able to predict the population of blast counts. This is, of course, a good example, but we found that this data, specifically the white blood counts, is very noisy. So we cannot predict correctly for all patients. So we are still struggling here to predict, to be able to correctly calibrate model to predict the population of these immune cells. And so we cannot predict here toxicity because the data is very noisy. So that's one problem that we are dealing with. But then, of course, we wanted to analyze the data further and try to understand under what circumstances we can expect the models to fit the data. And here we make one assumption, which is after treatment, the treatment lowers the neutrophil counts, and after treatment, the neutrophil counts should increase. So our hypothesis is that there should be a positive correlation between post-treatment duration and neutrophil counts. So this correlation can be computed from this data here, the population and the time after treatment. And here in this histogram, you see the correlations between neutrophil count, and post-treatment duration. And there are patients that have positive correlations, and there are patients with negative correlation. And those are interesting. We don't understand why patients can have lower neutrophil counts after treatment. But then we used this as a group in which we know that the immune cells recover after treatment. So we focus it on this group of patients. And as you can see in this plot shows kind of the error in prediction. This specifically is the fraction of unexplained variance for patients that are in this region, in the right region, and for patients that are in the left region, according to our hypothesis. And you see that for these patients, we can get better predictions. So we need to find out what's happening here for these patients, but at least the model can make relatively good predictions for these patients. And of course, once we calibrate it, we can use the parameters to see if there is some relationships between the parameters and clinical information. And what you see here is the value of one of the parameters. This parameter is related to how effectively the venetoclax is killing cancer cells. And these groups are groups that in which the patient responded to the therapy. But the last group are patients that are not responding to therapy. As you can clearly see, the values overall of this parameter are lower for patients that do not respond to therapy. And there are other relationships between these parameters and clinical features. Here is progression-free survival, which is the time after treatment in which the patient didn't get a relapse. That's progression-free survival. And you see, if we divided the patients in two groups, there is negative correlation of this clinical outcome and with this parameter. And for the other group, there is a positive or non-correlation between this clinical outcome or property and the parameter that we are focusing. And so we think that this indicates that the parameters have some predictive value, and that can be used in a further machine learning. We can use these parameters as features in a future machine learning to make predictions about outcomes of other clinical properties of patients. Finally, we are developing a dashboard, which is part of our digital twin, in which we are going to present all the data from patients, as well as data and predictions from models. This is still in development, but it is a fundamental part of our digital twin system. All right, I think that was all. Thank you for your attention. And if there are questions, I'm happy to answer.
Yeah, thank you very much, Aguilar, Boris. Any questions? Well,
I mean, I've got one I can ask. I suppose the obvious question is, how reproducible would the work be? Now, for the Boolean networks, you'll be using SPML, I assume, or something similar, because there are no standards for the multicellular modeling. So I'm not sure how much you can do to make it reproducible, even if you wanted to.
Because of the standards of the
Well, the way you
models.
represent, I mean, the way you represent the models, right? If somebody wanted to reuse part of it or extend it, if it's just blocks of code, that would be really, really difficult. But if it's in some standard modeling format, unfortunately, there aren't that many around. That makes those parts of the model reusable. Presumably, the genetic networks are in SPML. Is that right?
Since we are in the developing phase, we are still not using standards for model description or encoding the inputs yet. Or we have is codes in Python to generate these plots. I think it would be good for us to collaborate with the reproducibility center.
Yeah, no, sure. Yeah, no problem. I think we can do some areas can be standardized. Not all areas, because the community hasn't developed standards really for some things, although they're trying to now develop standards for multicellular models. Yeah, but anyway, yeah, we can talk more.
But that's surprising, because these models that I presented, the last ones, the ODE-based ones,
yep,
there are many of them for AML, for diabetes. There are so many. So I know it's not like the gene regulatory network models that are more standardized. But I think that I was surprised if there is no way to standardize this model.
I mean, if you have raw ODE equations, they can be converted back into their original biological interpretation. I mean, the idea is once it's modular, and using standards, you can plug and play and replace things with other models, and update models easily. And people could reuse parts of it in other digital twins. I mean, one reason why digital twins in industry has been successful is because they do they have a very modular approach to modeling. And they can plug in and plug out different components really easily. Like if they're developing an engine or something, or car manufacturing, they can plug in and out different components. And in fact, I think there are companies that specialize in building components that can be plugged into digital twins. And it'd be nice if, I mean, this is early days for digital twins in biology, but it'd be nice if eventually that would be the case for us too, where you have a model, so you have a basic model of a human, and then you can plug in or take away various parts to make it specific to a given disease or something. I think that would be really nice.
Yeah, that would be very nice.
Yeah.
Yeah, the difference in medicine is that the most, I guess, from our part, we are still focusing on making predictions that can be actionable.
Yeah.
That's our focus.
Oh, no, sure. No, that's really important.
But I understand that this is a collaborative effort for people more focusing on standardize it, and we should be collaborating.
I mean, there are certain models I know of that are very large, and I won't name them in public, that are completely non-understandable and non-reproducible. It can't be extended, reused, and an awful lot of resources have been spent in developing them, and they can't be reused by the community, which is a shame. So, but industry has a motivation to make things reusable, especially when they're dealing with large complex systems like car manufacturing or the aerospace industry, right? They want to try and make things as modular as possible so things can be reused, modified, update really quickly. So, hopefully eventually we will approach that level of usage too.
Mhm.
But it's early days.
Yeah, it's early days.
Mm.
Okay. All right. Let me stop the recording and start.